I just want to say a few things up front. So firstly, I was a scientist. I sort of set off on a startup journey, but I've sort of been superseded by Brendan Swain, who's sort of the man behind these clocks I'm going to talk about. So I'm sort of relegated to the second position as far as scientists is concerned. I know this is a very scientific audience. So Brendan will be there, you know, sort of saying follow up to answer some very deep questions. But I won't take I'll take as much credit as I deserve, not all of it, because Brandon's a big part of this. Just secondly, thanks everybody for showing up. I don't think we're very prolific in the literature so far, or sort of getting out there, but we're sort of hard at work moving things forwards. And it's just a real privilege to talk to this group. It's just, you know, like all of my favorites are in this group. You know, I won't name all the different people, but you know, it's a little bit, you get sort of starstruck sometimes by the people in this group. It's great to be a fly on the wall or sort of lurk in the background on some of the talks, but no chance I can do that today. So I will just get on with it now. So hopefully. Okay, so yeah, the title of my talk, drug target discovery for safer cellular rejuvenation by the application of driver clocks to cell reprogramming. So I've just got an image of some single cells on the right-hand side. And what we're doing is a very visual science, but this is actually a nice visual that we've got where we're organizing single cells to extract the underlying biology, which is a really interesting bit. And I'd just like to start sort of setting the scene. So not so long ago, we were talking about that, you know, let's do something about this. You know, how can we slow aging? But the world's moved on considerably. And now I call it really a race for rejuvenation, and just a friendly race for rejuvenation. And you know, I've just cherry picked 8. This is nowhere, you know, near comprehensive. This is almost just my favorites, right? So I'm not, you know, not writing history here. Anybody could choose their own 8. But I like these 8 for a couple of reasons. Firstly, that, you know, it's about age reversal. So it's not just slowing it down, stopping it. It's about bringing yourself back to a younger biological age, which is effectively age reversal. And you can sort of, you know, I can split this group down the middle. So one is blood-based rejuvenation, so changing the blood milieu to make it rejuvenative. And then there's this alternative paradigm, which is reprogramming, which is really basically cellular rejuvenation. The blood-based rejuvenation has seen the most inroads in the clinic, probably because it's so safe, because you're just playing around with the blood composition in most part. So, you know, a lot of this is going to be familiar to the audience. We'll just cover a few of it. So Greg Fahy, Intervene Immune, he reversed grim age by a couple of years. That was persistent, which was really exciting. And I think he's in a phase 2 trial with this cocktail, a couple of anti-diabetics and a growth factor. Tony Whisker-Ray is using altered blood plasma fraction. He's done things in Parkinson's. You know, the age-related diseases are much higher bar than rejuvenation itself. You've got the aging and you've got the unraveling on top of aging. So really impressive to see things going on there. Arena Comboy, basically translating therapeutic plasma exchange, super exciting. So that's a clinically approved therapy. Let's see what it does outside of its original indication. Howard Katcher in rats has shown you can reverse methylation age, which is our sort of best window into aging, so far by about 50%. Like that's 50%. It's not like an incremental amount of reversal. It's considerable, and you basically restore all of the organ functions to young levels. So I mean that's exciting. And then on the reprogramming side, which is you could argue maybe a more comprehensive type of rejuvenation, although you know it's like everybody's a winner here if these things work. So you know you've got David Sinclair. Basically, it's taking pluripotency factors, playing with the composition of the factors, so reducing it to RSK, reversing methylation biomarkers of age, restoring functions, which is a fantastic result. Turn Bio is doing great things. They've got an mRNA based system, slightly more factors, but you know that's comprehensive. And then you've got a couple of new efforts. Well, Calico is not so new, but the initiative is relatively new, which is trying to sort of go deeper into reprogramming. So let's look below the surface of what's going on, try and deconvolute the pathways. And a recent paper, they showed that just ONS was sufficient to rejuvenate and it had about half the impact on the identity of the cells, which is the big worry. Like, we want to rejuvenate, but we don't want to turn into a bag of stem cells in the process. That's no good for anybody. And then something that I'm excited about is, Joe Betts-LaCroix and Retro Biosciences. So I think I'm not trying to stalk their staff or anything, but everything that their staff is saying is very exciting, and it's like, it's coming from a great place. So apologies to the staff if you see me following you, but I'm excited about your company. So that's the race, and then why this discovery effort on cellular reprogramming? So why is more discovery necessary? Why don't we just go ahead with what we know? So I basically just call this like a powerful rejuvenation paradigm, but with risks, just with some risks. And how we mitigate those risks is, an area of active pursuit. So maybe just as a reminder, but in the top left graph, this is a data set from, I think, Yamanaka, and then Tamir Chandra at Edinburgh analyzed this data set using the DNA methylation clock, so Horvath's methylation clock, and basically showed, if you look at the focus on the blue line, which is methylation years or epigenetic age, you can take a cell from age 60 down to age 0 in just 17 days. So, 60 years to build up that aging and just 17 days to jettison it from the system. So, about as rapid a rejuvenation as we have come across so far. And the exciting thing is, up until about 15 days, these cells still retain their original identity, not 100%, but they've still got a memory of it and can revert back to their identity. So there's a sort of zone where you can rejuvenate but not become a stem cell. So that's according to the clock, but that's just a biomarker. If you express a subset of these Yamanaka factors in the optic nerve of aged mice, you can restore a sort of remarkable ability that only newborn mice have. So basically, newborn mice, if you crush their optic nerve, a bit of a barbaric essay, but if you crush their optic nerve, it degenerates. And a newborn mouse can basically regenerate that optic nerve. After just a few days, that mouse loses the ability to regenerate its optic nerve. However, if you put the factors back in to the mouse and express them, basically an aged mouse can now regenerate its optic nerve just like a newborn mouse. So you regain these functions. So that's quite a dramatic result, about as a dramatic functional result as you can come across. And then just on the right hand side, this graph, you can increase lifespan in mice, but you have to be in this Goldilocks zone. Not too little because nothing happens, not too much because you get these pluripotent teratomas, sort of scary cancers. But if you're just in the middle, which is basically partial reprogramming or just pulsed reprogramming, you get a nice lifespan extension in progeroid mice in this case. I'm sure there'll be some interesting follow-up studies to come. But anyway, this is basically a very promising paradigm, but these are pluripotency factors. Let's not forget that. They're designed to make things into stem cells, and that is, sort of it's the direction of cancer, right? The direction of stem cells. So we need to proceed with care. So if we do something in one system, will it necessarily work in the other? We don't know. So it'd be great to increase the safety window, like not worry about playing with fire, so to speak, or walking the tightrope. So where do we fit into this? Because I didn't start out interested in cellular reprogramming. So I'll just try and tell you sort of high level of the story, and then we can sort of unwrap it a little bit. So yeah, just the high level. We've identified, this is where we are now. We've identified some putative drug targets for safer cellular rejuvenation. Basically by applying machine learning driver clocks to cell reprogramming. So machine learning, we aren't the first people to arrive at machine learning, but I think it's our application to cell reprogramming we're excited about. And we're basically ready to validate these targets. We've got this list of genes that we want to throw into a cell, and now it's just a matter of just bringing in slightly more financing and then testing these genes and seeing if the bioinformatics is as exciting as we think it is. So let's just sort of go back to the beginning, just of the startup journey. So we set out in 2017 with the backing of Jonathan Milner, who's an angel in Cambridge, UK, basically to tackle mitochondrial DNA mutations. So this is a type of damage that accumulates with age. And we had sort of some promising sort of tool molecules that could basically combat these mitochondrial DNA mutations. Something I wanted to sort of look at early on was like, can we show that in a sort of an unarguable way that this is the right approach to therapeutic intervention and aging. And it was actually my investor Jonathan that put me in the direction of the epigenetic aging clocks. He said, "Oh, have you heard about these aging clocks?" And I hadn't, right, because I was a little bit blinkered. It was like, mitochondria was my world. But the moment I found out about these clocks, it presented a fantastic opportunity to audit our hypothesis. So if mitochondrial DNA mutations were really, the be all and end all of aging, then they should have a dramatic effect on this epigenetic aging clock, which was sort of an agreed upon measure of aging. I know there's lots of measures of aging, but this was relatively new as far as something everybody could agree on. So we'd done a mouse experiment with this mouse called the mutator mouse. Basically, it has way more mitochondrial DNA mutations, and it gets older faster. It gets premature aging. So it's very provocative. The link between this damage and the aging phenotype. And we had a drug that we could basically slow down some of these aging phenotypes. So there were two questions. Does this mouse, which shows a premature aging phenotype, show acceleration of the clock? Is there this really strong link between mutations and the clock? That's the first question. Second question was, does our drug impact the clock? So there was no such thing as a mouse clock service that we could send samples off for measurements, but Wolfram could just sort of define the first multi-tissue mouse epigenetic clock, because these were mouse experiments. I couldn't use the original Horvath clock. But yeah, Wolf didn't have any spare hands. So what we did was we sort of recruited Romina, our sort of first full-time employee, and we embedded her in Wolf's lab, basically to learn the method, so that we weren't taking up any bandwidth, but just doing what we needed to do. And we measured the clock in our mice, and surprisingly, the mutator mouse, although it showed a premature aging phenotype on the outside, the clock wasn't accelerated at all. So the clock was just the age of the mouse, chronological age of the mouse, even though the mouse looked dramatically older. So that was a surprise to us. Even more surprising was that we slowed down the epigenetic aging clock in these mice, right? So there didn't seem to be a connection between the clock and the mutations, but a drug designed to reduce the mutations, slow down the clock. We later found out that only in this mouse do we slow down the clock. If we use these drugs in wild type mice, we don't slow down the clock. So there's some interesting biology there. But the main thing about this experiment was that we were trying to face the music. Is this the best lever for therapeutic intervention in aging? And certainly it's an interesting approach for a niche aspect of aging. But for us, it was a pivot point. It was like, okay, the clocks told us this isn't perhaps the best approach right now. Where do we go to look for better levers? And I think around the same time, we took on an intern called Brendan Swain, who I'll talk about a lot more later. And we were really excited about clocks, number one, but also the Tabula Muris Senis, which was this fantastic sort of aging resource. For those that aren't familiar, the Tabula Muris Senis is a database with sort of single cell transcriptomes of the aging mouse, so sort of multiple time points in the aging mouse from every single cell and every organ in the body. It's basically a sandpit or playground for aging scientists to, sort of try and come up with things and look for phenomenon. And what me and Brandon were really excited about is basically, looking at this and looking at this sort of data, this data set. But secondly, the logical extension of aging clocks, which is, could we get a clock and do a CRISPR screen for aging? So could we knock out every gene in the genome, have a clock to basically get a readout and get a relationship between every gene in the genome and aging? So sort of systematically go through everything and let the data do the talking. Like the genes will reveal themselves according to their relationship to the clock. And the technical solution that we needed to do this CRISPR screen for aging was a single cell aging clock based on the single cell transcriptome. This was the enabling technology. So we basically played around with this Tabula Muris Senis, trying to develop methods for accurate single cell aging clocks. And the short story is we did manage to crack this problem, like generate accurate aging clocks with single cell transcriptomes. They have advantages, they also have disadvantages, but we did crack this problem. I think just before we were going to go ahead and do the CRISPR screen, with this clock as a readout, we realized there were a couple of problems. Firstly, a CRISPR screen, the screen we were planning would just be single CRISPR knockouts. So on a 10x genomics machine, you can do a single CRISPR, knockout in each cell and get a simultaneous transcriptome from each cell. But that's only single gene perturbation. So you basically limit your question to how can I rejuvenate with a single gene, which isn't the more exciting question is how can I rejuvenate, like whether that's a combination of 4 genes or 5 genes, we don't know. So the 10x system that we were going to use, we didn't have access to combinatorial space. So that was the first problem. And then secondly, we realized that the clock we generated, it was built out of genes. And these genes were very interesting, right? Some of them were known aging genes. And the more we looked at them, the more we realized we were looking at a lot more than just a single cell aging clock. We were perhaps looking at the aging biology itself. I mean, the original epigenetic aging clocks, it's a big question, like what are they? What's the underlying biology? But with these gene-based clocks, those answers were easier to sort of spot. So now we're at the Jeffrey Chi Biomedical Center in the Milner Therapeutics Institute, which is Jonathan's institute. Not everybody's got an institute, but Jonathan does. And we're basically going to try and validate these genes. So that's a high-level story. And just a bit of detail on the technical breakthrough. So I have to talk in sort of simplified terms. But what we did was we took a sort of a gene expression matrix, basically gene expression from single cells, and we basically do a dimensionality reduction. So basically, we go from genes to pathways, and this distills the information relevant to a time course. So there's aging time courses, there's rejuvenation time courses. For instance, when you express Yamanaka factors, that's a time course of rejuvenation. There's also time courses for age-linked diseases, which have an aging dimension and have a disease-specific dimension. So this method is applicable to all of those scenarios, but we focus on aging and rejuvenation. So we did a dimensionality reduction, and we basically distill the information that's interesting in that time course. And then secondly, we basically use these pathways as input for a machine learning model of the time course, so aging clocks basically, and the same sorts of things, elastic regression. And then we basically have this model, this machine learning model, and this retains information about the contribution of individual genes to the model. So it's like a ranked list of genes. This gene contributes the most, and then this one and this one, and that ranking is key. It allows us to prioritize certain biology over other biology. And just some advantages of these gene clocks, because I think we realize this after the fact. So gene clocks, firstly, they allow you to enrich driver biology from a time course, just as evidenced, right? So on the left is basically it's the relative weightings of certain genes in a clock trained on human fibroblast aging. And you see mitochondrial genes show up, you see ribosomal genes show up, you see like a gene that's sufficient on its own to drive progeria or accelerate aging on its own. Like these are known drivers of aging or been linked to aging. So it's really interesting to see. It doesn't necessarily mean that we're better than another clock, but it's encouraging to see these things. The second thing about gene-based clocks is that you can rapidly test causality with mature technologies like CRISPR or overexpression. So you can knock out a gene, activate a gene with CRISPR, or you can overexpress a gene with CRISPR and see if you affect aging according to lots of different measures. And also genes are very easy to target therapeutically. So you can have a therapeutic mRNA, or you can develop a molecule targeting the gene product. And the last thing is we got the single cell resolution. That was the reason we created the clock, to try and get this single cell resolution, but it turns out it's more of an academic interest than anything else. And then just show you some of the things we've done with this clock. So this is basically a single cell RNA-seq time course of aging fibroblasts. So we've only got 5 time points in this left-hand graph, but we've basically got predicted age versus actual age. So we've got a couple of samples towards the lower end, and then a couple of samples towards the higher end, and then sort of a middle-aged person in the middle around 50. But you can see there's individual dots, so we can predict the age of individual cells with a Spearman's correlation coefficient of 0.94. So that's per cell. Interestingly, the samples that are older, like one person seems to have aged a bit faster than the other. I'm sure there's an interesting story there, but that's interesting to see. And then when we look at the genes that make up this clock for aging fibroblasts, we see these genes show up which have been linked to aging before, which is really encouraging. But like I said, this method could be applied to any time course. I guess aging is just it was just such an area of interest for us. That's where we went to look. But if you take Yamanaka's public data, you can basically train a clock on rejuvenation. So actual rejuvenation means DNA methylation based readout of rejuvenation. So we basically train our predictor against that DNA methylation readout. We get a really accurate clock, 0.95 Spearman's correlation coefficient. And then we get a bunch of genes, and these genes are really exciting because they don't look like pluripotency genes. A lot of them look very familiar, like from the aging side. And we're particularly interested, I've just labeled a couple of genes because of their sort of expression trajectory. So gene 1, it basically, it starts at a low level of expression, and very quickly, like in the first 3 days of the rejuvenation time course where OSKM, the Yamanaka factors are expressed, it peaks and then it sustains. So this is very similar to the rejuvenation behavior. You don't have any rejuvenation for the first 3 days with Yamanaka factors, and then you get sustained rejuvenation. So we're very interested in the type of genes that map to this rejuvenation pattern. And then gene 2 is the opposite. So there's a gene with high expression, it dips quickly and then it stays down. So what we're trying to do now is we've got this list of targets, and we basically just want to extract combinations. So we're going to extract lots of different combinations. So there'll be non-rejuvenating gene combinations, so it's basically genes that are neutral. So our clock arrives at these genes, but they're just it's just like fat. It's just fat, doesn't really do anything. But there are some gems amongst the fat. Not a very good analogy, but I think you know what I mean. And then we're gonna also find pluripotent. Basically we're gonna find pluripotent combinations. So we want to move past this because that's where we are with the Yamanaka factors. But we expect to see basically combinations of pluripotent and rejuvenating genes. But this is what we're really setting out to do here: finding combinations of non-pluripotent and rejuvenating gene combinations, and then also the interface between all the other genes, really trying to separate the rejuvenation part of reprogramming from the pluripotency part. So this is a gross oversimplification. There is a more detailed slide, but this is the idea, right? We have this list, and now we're throwing these into a cell system where we can rapidly validate them. This is our roadmap, just a very high level. So we've created this gene driver clock originally to do a CRISPR screen for aging, but there's much more we can do with it. We've just protected this, which makes this talk much easier. What we want to do next, which is this is a funding round we're sort of about to open. We're going to distill the minimal set of genes sufficient to safely rejuvenate a human cell type. So, we don't know what we're going to arrive at up front. Will we move beyond the Yamanaka factors? Will there be slightly safer, or will we actually be able to fully separate pluripotency from rejuvenation? But the excitement is we can see these genes, and we have a confidence that we can fully separate. And there are others like Calico that have shown that you can start to separate, but they haven't sort of exhausted the path ahead. So once we arrive at that sort of minimal gene set, we'd like to then basically it's a big IP milestone, so we file some IP, and then the real work begins, which is we've got to start basically lead development. So for a general purpose rejuvenation therapeutic, we develop that ourselves, but if there's a particular disease indication, we don't want to have to build a specialist pharma company again, so we'd probably partner with pharma for that one. But that's the next stage, so one step at a time. Just some of the characters that have been roped into this. So myself, I originally had a training in mitochondrial biology, but then through the sort of pursuit of rejuvenation, not just mitochondrial rejuvenation, I've ended up in a very different place. Brendan was an intern with us in 2018. Sort of just we maintained a conversation, and this is basically the direction we're going now. It's Brendan's doing, so he deserves most of the credit. Romina, I worked with when I was a mitochondrial biologist. We brought her in so we could put her in Wolf Reich's lab and do these first COP measurements. Steve Ives, not my brother, he is my dad. He's not taking our rejuvenation drugs. He does look quite young. So he was a serial entrepreneur. So he's just, if you've got that card to play, you've got to involve that person. He just saved us a lot effort and dead ends and making stupid mistakes. So I'd be crazy not to turn on him, although it's complicated, right? You've got a family relationship and the work relationship. And then we are recruiting a number of positions. Once we've got a bit more money, we can recruit all of those, but we are recruiting at least one lab scientist right now. Just because we just need to see if it's going to work or not, right? It's like, now's the time. These are some of our advisors. So Jonathan Milner is a fantastic angel based in Cambridge, UK. He's founded many very interesting companies. Abcam was his baby. More recently, Foremost, Helix. Anyway, you can look him up. Ken Raj has been a co-author on many of the landmark epigenetic aging clock studies. Ken's a fantastic guy, sort of real diamond. And he's given us some fantastic advice, over a period of time. Wolfreich let us into his lab very generously, and he's doing some really interesting things in aging, particularly multi-omics. So at the moment, the aging clocks are confined to one omic layer, but if you've got multi-omics, you can train clocks across layers. Starts to get crazy, but in a good way. Aubrey's been really supportive, fantastic from a long time ago. Very busy these days, but yeah, he's great. And then David Billington, he's got some serious experience in sort of big pharma companies, just make sure we've got our head screwed on. And now just some supporters. So Milner Therapeutics Institute, where I'm based, it's sort of like an accelerating environment. It's not necessarily an accelerator, but it's like working in a big institute, but we're a small company embedded inside, so we can just do things faster. We're basically here for this all hands on deck experiment. I'm really pleased that Alison accepted us as an accelerator company. We have been going sort of for 4 years, but we're very small, so we're only 3 full-time employees. I think all the help we can get, to be honest, that's a good thing. And we were part of the Creative Destruction Labs, which is like almost like a virtual Y Combinator, a Canadian-based effort. And I was actually put in touch with somebody that's been very valuable to us. Accelerate Babram, that was basically Babram's the site that Wolfreich was at, and we went on this accelerator. We got a nice big check, and we got lab space and things like that. And so that's basically us. And then I think because I'm talking to this group, and the abilities of this group are so large, it's a good place to ask for technical solutions. So if anybody's working on these technical solutions, this would also help accelerate things. So I don't know if any of these things are possible, but if you don't ask, you don't get. So the first thing is it's basically a skin and blood clock, but for biological age. So there's something called the skin and blood clock at the moment, but it's for chronological age. So basically you can measure aging in fibroblasts, and that also tracks aging in the blood. So it's sort of there's a connection between what's going on in vitro in your dish and what's going on in real humans. But that's just for chronological age. And if we want to develop therapeutics, they're going to have to target diseases. And so what we would really like is something like a grim age that we can use in a dish, or a systems age from Morgan Levine that we can use in a dish. So when we're testing things in a dish, we can say, oh, look, our time to heart disease is going up in response to this perturbation. We'd like to know as soon as possible. So that would just be fantastic. It would like de-risk the project substantially. We could show that we're actually like pushing away with a prophylactic intervention, we're pushing away age-linked diseases. Secondly, maybe a little bit simpler, a biological age clock for mice. So if you could track like pathological age-related changes, and you basically train a clock including those changes and chronological age, so we could basically test our interventions and do those mice move further away from age-linked pathologies. Again, it just helps de-risk things. Because grim age, you'd have to do human studies, and systems age, you'd have to do human studies, and we can't just teleport to human studies. We've got to get there. Thirdly, once we're in human studies, or even these biological age clocks, if they were disease optimized, so if there was like a grim age for heart disease that was optimized, so it gave us the best possible resolution, that would be great. If we were targeting a certain disease, having a clock corresponding to that disease would be great. Obviously, we can have that in a mouse, even better. We can have that in a dish, even better. Number 4, I know that you only get 3 wishes, but number 4, single cell multi-omics. So if we could just basically do multi-omics for a time course, whether that's aging or rejuvenation, and train a clock that basically picks out the best biology, whether it's a CPG site, whether it's a gene, whether it's a chromatin accessibility, we just, we're able to sort of cherry pick the best features, not just the layer that we've decided to use, whether that's a transcriptome or something else. So if anybody can come up with some good multi-omics methods, that would be great. I think Wolf Reich has developed something called SC-NMT-seq. I won't elaborate the acronym, but I think that measures 3 molecular layers, and they've got plans for 4 and 5. I really think that's going to be fantastic. And then lastly, basically just data sets for age-linked disease time courses. So, there's a relative abundance. I know it's relative, but there's an abundance of aging time courses and rejuvenation time courses, but age-linked disease time courses are a little bit harder to come by. But when you've got these methodologies, you can train them on the age-linked disease. So you can potentially pull out the driving biology for the disease-specific side and also the aging side. And you put those two together, you've really got something interesting. If you want to get hold of me, my email address is Daniel at shiftbioscience.com. I'm going to be on SwapCard at the scheduled time. I'll be trying that for the first time. Sorry if it's sorry if I don't get the hang of that fast enough. That's the address if you don't have it. Also, just an extra bonus engagement. Tomorrow, me and Brendan and some others have been playing around with something called Pluto. So basically, it's like a virtual space, but it's like Zoom, yeah. So you've sort of got your face, but on this cube, and you can like walk around as this cube, and you can talk to each other in this virtual space. And like we put our aging clock on the wall, so we could talk about that a bit more. But basically, if you've got a browser, you can just use it. It's pretty good. I've only got it to work with Safari, so there might be some gremlins, yeah. So sorry about that, but it's a lot of fun. Me and Brandon are desperately trying to maintain a professional face here because we're having way too much fun with this. But we'll be there from 8 a.m. Pacific time tomorrow, which is 4 p.m. our time. If you want to have like a sort of high feedback discussion outside of the constraints of SwapCard. And that's it. I've just got some appendix slides, but I'll only go to those if we need to.
Fantastic. Okay. Well, thank you so much. That was much more than I expected. This is awesome. I think maybe just remind people in the chat when they can catch you tomorrow. I'm going to remind them about the SwapCard. So just that people already know how to follow up if what we're discussing now is not enough. But let's move to the Q&A. I think Nir had his first question, and then we will go with Shish.
Daniel, first of all, that was really terrific. Everything about it, the ID and the preliminary data, and you're terrific, and congratulations. I've really two kind of questions there. The first one is, am I understanding right that you're working from data on mice and on fibroblasts that are taken from mice? And if so, why not humans? And when you answer, I'll tell you why I'm asking.
Sure. So the data I presented was actually data from human fibroblasts, and yeah, so there was an aging time course, and that was human fibroblasts, so human subjects. I think there were a couple it was polarized, yeah. So there were a couple that were young, one in the middle, and then a couple that were sort of 70 years old, but they were humans. And the rejuvenation time course was human fibroblasts. So I think Yamanaka basically they were age 60 and took them back. But we have done this on mice as well, and it shows similar results. So we did all of the experimenting to create the method on the tabula senis, but then we applied it to human cells, and we see the really exciting stuff in the human cells.
So the reason I'm asking is that I have data that you'll understand I haven't published, where we took humans and gave them metformin and placebo, or acarbose and placebo, ITP drugs, and it was a crossover study, but we did the same in animals. So we have transcript from humans and from animals treated for the same time, equivalent dose of drugs, and to our surprise, the transcripts are not the same. Although the upstream regulators are the same, okay, but the transcripts are not the same. So I think we have to be careful how we go from human to animals, and of course, that the clocks are not the same, right? So I think I think from a drug development, you should really think only on humans, right? In this sense, otherwise you might be confused with the experiments. The second question for you is, are you putting some positive controls? What I mean is, do you put to those fibroblasts, do you treat them with metformin or rapamycin or NAD or something that actually has effect on epigenetics, but actually to get the land field of what you're doing in relationship to gerotherapeutics?
Yeah, I think that's an obvious thing to do. I don't think we've done that so far, which is like, let's look at like, you know, gold standard interventions, like what does rapamycin do to this clock? Yeah, and if it does the expected things, the clock's meaningful or more meaningful. Yeah, I think that's an obvious thing we can do that we haven't done.
And what it doesn't do, right? I mean,
what it doesn't do.
Both things. You want to know if you're adding to anything now, and is it important?
Yeah, that's a very good point.
Terrific. Thank you.
Thanks, Noah. Thank
you. More work carved out for you, Daniel.
It's great work.
Yep. Okay, well, Carl had a point that's directly, I think, piggybacking on you. So Carl, do you just want to go before we move further down the question?
I'm not sure what point you're thinking about, because I just hit enter on my point responding to Nir. So the nice thing about this platform with regard to humans versus mice is that they should be able to look at both sets of clocks and actually limit to things that work in both. And of course, we care about them working in humans, but if we go down, you know, if they go down a clinical path, then working in mice is a good thing if it's the same thing that can work in both. But the platform should allow filtering, you know, if there are enough leads, should allow filtering down to things that work in both.
Okay, good. We'll leave it at this, or any comebacks? All right, then next one up, we have Ashish.
Hi, Daniel. Very nice talk. I also agree with Nir. If we can have like human data, it would be really nice. I have two questions to you. So as you know, single cell sequencing is a little biased towards less expressed genes, such as transcription factor, which is really important for aging and pathway generation. So how did you solve this problem? And my second question will be, how are you doing like whole methylome analysis for identifying cell biological age, which is coupled with CRISPR?
Okay, so the first question, I'm gonna defer that to Brendan because that's very specific. But with respect to the rejuvenation time course, it wasn't actually single cell data in the rejuvenation time course. It was cells that were sorted for a cell surface marker of pluripotency. So it's actually bulk measurements, but we can use the same method on bulk measurements. So we don't have this, like you said, in single cell data, you have this coverage problem. And the rejuvenation side, we don't have to worry so much about that. For your first, yeah, for the single cell data, I will have to defer to Brendan for that one. So yeah, let me write that down, and I'll write your name down as well.
Well, I can answer that now if you want.
Brendan's on the call. Yep, that's great.
So I think I don't want to reveal too much about the method, just because of IP concerns. But basically, the method isn't just based on the absolute variance of genes, which would be the major contributor to that problem of picking out genes that are the most variable across the data set, which is kind of how a lot of this kind of analysis is done. But we've basically made great efforts to basically allow genes to contribute equally to the clock and make sure that we're not favoring the loudest shouters, essentially.
Mm-hmm. Yeah, I'm asking because yeah.
Sorry, Ashish, could you repeat your second question so I've got that ready?
My second question was like, are you doing whole methylome analysis for identifying cell biological age, or are you doing a specific methylation signature?
So we've started on the transcriptome, but I think I sort of talked about multiomics, where if there were multiomics, basically we were at the mercy of the public data sets to begin with, the tabular cenis, aging time courses in human cells. And we're just starting to generate our own bespoke data sets for the systems that we're going to be using to sort of try and validate our genes. So yeah, we'd be looking to do beyond transcriptomics and when we start doing things ourselves, but in the basically early days, we were at the mercy of the public data set. So I don't think there was anything there at the time. I think, did you say metabolomics? We weren't able to use that.
Okay, thanks.
You're welcome.
Lovely. We have Yoti next. I will ask it for you if you don't mind. Her question was, is all the data limited to fibroblasts?
No. So in the tabula muracenis, which is mouse, let's just qualify that as mouse, you've got a lot of different cell types. Certain cell types have richer time courses. So as far as using our clocks is concerned, we're better off with a richer time course. But on the human side, on the rejuvenation side as well of humans, I think there's reprogramming paradigms from 5 different cell types. So reprogram from endothelial cells, reprogram from like 4 other cell types, and you can train multi-tissue rejuvenation clocks across all of those and see what the biology is. Because obviously, if it works across lots of tissues, the translation is the potential is higher from a translation perspective. So yeah, not just fibroblasts. You're not just gonna be have really great young fibroblasts, and the rest of your body falls apart around your fibroblasts. And we are shooting a bit higher.
Okay, lovely. Then I know that you've already came up with a wish list here for this group, but just to really be quite specific about like what would actually really help you move this project forward now. And if anyone here is on the call that, or even afterwards for those that will watch it on YouTube, like what are specific ways in which they can reach out to you, as making it as actionable as possible, perhaps is a good way to go.
Yeah, so beyond the wish list, which is maybe a little bit further in the future, some of those things, immediately we just want to test these genes as fast as possible, and we're doing a lot of these things for the first time. So we're just looking for people like preferably in Cambridge, UK, that can help us. We need a lab scientist at a very minimum. We are raising a certain amount of capital. It's not like a ridiculous amount, but it's a lot more than we've raised before. So if there's anybody out there that's very passionate about clocks, trying to find, sort of go beyond Yamanaka factors, explore the unknown, see what's there. There's so much that we don't know, okay, and it's exciting to have an approach where we can reach some of that. If any of that sounds exciting to you and you want to help move that forwards, then please get in touch. Really, it's just we need, the support now would be so much more appreciated than say in 6 months' time. Like we really need to get going now, because they're just sitting on the list, right? It's just be such a tragedy they were sitting on that list for any longer than necessary. So we just want to start testing it.
Okay, great. You even included the urgency in it. That's awesome. Yeah. Okay, maybe and a little and anyone else who has a more specific question, please stop me. Otherwise, I will just go and ask a few more general questions about where do you see, if this is successful, where do you actually see it? I mean, you already talked a little bit about immediate next steps, but where do you see potentially this going in like 5 to 10 years? Like, are there any really kind of like very, very high hanging fruits that you hope to be able to eventually get to?
Yeah, so I just up front, drug development's a very long and painstaking process. So you've got to set expectations, but at the same time, the whole COVID response, the goalposts were moved for that. I've got a feeling that if there was something really exciting and safe moving forwards, there'd be a certain amount of energy pushing that forwards beyond expectations. So I don't know, in 10 years' time, I'd expect something to be late clinical stage, but there's so many steps in between, right? And I think getting it right is more important than timeline, but obviously the faster the better, because if you get these things to work, we'd rather have these things sooner. I think even before that, it would be really great just when we found the list of safe rejuvenation genes to engineer sort of a mouse embryonic stem cell that you can just induce these genes. So you don't have to worry about sort of all the getting drugs to every part of the body, but just induce it in a mouse embryonic stem cell, induce them, and then just watch the clocks, like do the clocks dip and go up, and can you just basically reset to infinity? Is that the first mouse where you can, that would be a really eye-opening you know, just spectacle, yeah, to see what would happen there. So I think generating that mouse embryonic stem cell would be a really exciting thing that isn't so far in the future, but you'd have to give that a bit of time, right, for that mouse to basically live. There'll be people that aren't specialists or that aren't enthusiasts, and they'd be waiting for, you know, the mouse to just live longer. Yeah.
Exciting. And you already talked a little bit about the race, you know, at the beginning of the talk. And do you have any ideas and, you know, potential collaborations going forward, even with other folks in that race? Because I think what's so exciting about you, and I guess Nathan, who's also on this call, is that, you know, you're really trying to pull the whole ecosystem up, and like, you know, you've been doing quite a lot of work on this. So I think it's really interesting how you position yourself really in this ecosystem of like a rising tide.
Yeah, I mean, I think Steve Horvath's Clock Foundation, like just developing these clocks before, you know, to basically speed the drug development. That's great. I think Morgan Levine's got some really cool stuff, like enhancing the clocks. That's an obvious one. I think it's very hard for me to sort of analyze what's out there and sort of try and connect that to what I'm doing. So if you think you can connect to what you're doing, obviously I'm going to take the time to listen to that and see if that is you know, that is a good fit. So yeah, I mean, I'll keep an open door, and it served me well.
Lovely. Well, we do have, I think, Morgan on this call. I'm not sure if Steve's on it right now, but if that tickles your interest, then maybe you can reach out to Daniel too. All right, lovely. I think Carl made a comment. Do you want to make it here in? No. Okay. Well, then maybe you could look at it in the chat. And you know, do you think that there's any you know, potential, yeah, any potential risks or downsides that you see, like on the, like, let's say, you know, any potential reasons why this could not work that you know, you could like really with which you could falsify those in the next few years or so? Like, is there anything that keeping you up at night? I mean, you're already testing this, right? But is there anything else where you're like, okay, well, this would really be a potential bottleneck that we need to overcome, that, you know, maybe people here in this call could even advise on?
Yeah, I think the thing that keeps me up at night is translatability. So we could get very excited about what we're doing in our specific system, but then we don't have a connection to all the other systems. So I guess Steve Horvath's mammalian clock is a really great way to try and dispel some of these fears, you know, because it's connected to so many different contexts of aging. So when you move that, you know, you've really moved something. So that's one way to do it. But yeah, it's just making sure we're not system specific. So if we do find a minimal set of safe rejuvenation genes, we just want to, you know, broaden from our first cell type to multiple cell types, make sure that works in mice. We've got to go through mice to get to humans, whether or not we like it. Maybe organoids, you know, all of the systems. Yeah, just make sure there's no reason to go narrow, yeah, and sort of try and save pennies when we could just sort of increase the breadth and translatability. So I think translatability is the biggest worry. And then the safety side, yeah, can we fully deconvolute from pluripotency? So that's not a foregone conclusion, right? So we need to do the study. So there's a discovery element to this, but there's a lot of promise just from what we've done so far. So yeah, the opportunity's there, right? And we just got to make the most of it.